Hi friends, welcome to Arnav. Today is the 8th episode in the biology series. So today we are going to discuss about the lysosome. So it is a membrane-bound organelle found in many animal cells. It is found in many animal cells, in eukaryotes mainly. There are spherical vesicles that contain hydrolytic enzymes that can break down many kinds of biomolecules. Like these enzymes are used in the stomach to digest foods, so we can get energy from those foods. That's why this lysosome can break down stuff like useless things. Like there are proteins which are already used. A lysosome has a specific composition of its membrane proteins and luminal proteins. Yes, there's a specific composition. Specific ones. So the lumen's pH is 4.5 to 5. It is optimal for the enzymes involved in hydrolysis. Lysosomes were discovered and named by Belgian biologist Christian de Dieu. It was discovered in the 20th century. He received the Nobel Prize in Medicine for this discovery in 1974. 1974. A bit recent. Lysosomes are involved in degradation of polymers, secretion, plasma membrane repair. Yes, they are also repairers, even if they are destructors also. And apoptosis, which is the process where a cell kills itself. For example, there's a cancer in outbreak and suddenly the cell got the cancer. It was the first one. There was a small mutation in it. It will get cancer, but it killed itself. So there's no cancer anymore. That's the use of lysosomes. Because of this lysosome only, there's no cancer in us everywhere. Just we are not cancer anymore because of lysosomes only. So cell signaling and energy metabolism are its other jobs also. So lysosomes act as the waste disposal system of the cell by digesting used materials in the cytoplasm from both inside and outside the cell. It takes used materials like proteins which are already used. Material from outside the cell are taken up through endocytosis, a process where a small vesicle forms from outside the cell membrane that means from the cell membrane to go inside and through the lysosome. And while material from the inside of the cell is digested through autophagy, where the vesicle goes into the lysosome and mix with it, and the item will get burnt. It will be broken up. Lysosomes also acts as a safeguard in preventing pathogens from being able to reach the cytoplasm before degraded. That means when we are ill, there will be special immune cells. These immune cells will eat the pathogen like bacteria. The bacteria which is attacking us, not the bacteria inside their body which evolved to our environment. Those unfriendly bacteria will be eaten by the lysosomes in special killing cells called macrophages, which eat the pathogens, or even viruses also. So lysosomes are known to contain more than 60 different types of enzymes and have more than 50 different types of membrane proteins. Different proteins for just lysosomes. Yes, dustbins. Wow. So enzymes of lysosomes are synthesized in the rough endoplasmic reticulum and is exported to the Golgi apparatus where it is processed, and then a vesicle will take it and will turn into a lysosome. Eventually will turn into a lysosome. Lysosomes are involved in a group of genetically inherited deficiencies or mutations called lysosomal storage diseases, or also known as LSD. So these genetic deficiencies are related to several neurodegenerative disorders, cancers. Yes, the one taking out cancer will also be the reason for some types of cancer. Yeah, cardiovascular diseases and aging related diseases. Yeah, so now I will explain the process of endocytosis in a diagram. So first, in the extracellular matrix, which is the place outside the cell, between the cells, there will be a food particle. For example, here is a food particle, but normally there could be any other particles. So this green thing is the food particle. So there is a cell membrane. When the food particles gets closer to the cell membrane will wrap around it to form a type of vesicle called the food vacuole. Its membranes are made of the cell membrane, not the normal vesicle membrane. So the food vacuole will go deeper into the cytoplasm, into the cell. So next, a lysosome with hydrolyzing enzymes will get closer. So when it gets more closer, it touches it, where it fuses. So the lysosome fuses with the food vacuole and releases the enzymes into it. It just injects it, like here. So then the hydrolyzing enzymes digest the food particles, like here. These red dots are the hydrolyzing enzymes, and these green dots are the food particles, which are now broken down into smaller pieces. Yeah, this is the process of endocytosis. And wait a second, the process is not fully complete yet. If the item is useful, the food particle, here is the example. If it is useful, it will not fully be broken down. It will be again recycled. But if it is not useful, it will be broken down to the atomic level. So this is for today about the lysosome. Bye.